Welcome to my channel Daily Shop. In this video you will learn how to perform gram staining. Gram staining is a common technique used to differentiate two large groups of bacteria based on the different cell wall constituents. The gram stain procedure distinguishes between gram-positive and gram-negative groups by coloring these cells red or violet. This technique utilizes a 4-step staining procedure with 2 different dyes and is one of the first tests used when trying to identify unknown species of bacteria. Arranged Bunsen, loop, dyes, slide, wax pencil and sample. Using a wax pencil, label a microscope slide with your initials. Then draw a circle about 1 centimeter in diameter on the slide. Using a metal inoculation loop, sterilize it by flaming. Using aseptic technique at a drop of sterile water so that a film appears across the loop. Transfer the water into the circle drawn by the wax pencil. Flame the metal loop again to sterilize after allowing it to cool. Use the metal loop to transfer a colony of bacteria growing on an agar plate. Make sure to lightly touch the bacterial colony to avoid transferring too many bacteria. When finished, flame the loop to sterilize. Let the liquid completely evaporate, leaving behind a dry spot on the slide. This will take at least 10 minutes. Once the liquid has dried completely, heat fix the bacteria to the slide by quickly passing this light through the flame 3 times. The flame should touch this slide for about 1 second during each pass. Ensure that all Bunsen burners are turned off before obtaining the set of gram stains. The decolorizer contains a high percentage of alcohol and it's very flammable, and a few drops of crystal violet stain to the bacteria into the wax circle is filled with stain. Let the stand for 1 minute. Rinse the slide by dipping it in a beaker of water 2 to 3 times until most of the stain has been washed off. Next, add a few drops of grams iodine to cover the bacteria and then let this stand for 1 minute. Rinse the slide in a beaker of water until most of the iodine is gone. A second beaker of water can be used to ensure most of the iodine has been rinsed off. Now add a few drops of decolorizer. Let the decolorizer flow over the bacteria at a 45-degree angle until the flow is clear. Then finished by rinsing the slide in a beaker of water. Counters gain by adding suffering and stain to cover the bacteria and let it stand for 1 minute. After 1 minute, completely rinse the slide in water. Allows the slide to dry completely. Once the slide is completely dry, observe the sample under a microscope. For observing by microscope, use tissue to make it dry. Then set the slide in microscope with flushing light and adjust by moving platform. Left picture identify the gram-positive bacteria and right on a gram-negative bacteria. Thanks for watching. Please stay with us by subscribe my channel.